There are three different parts to interphase. First up, growth 1. In this phase, the cell does most of its growing. It does its normal job, which may be being a heart cell, a lung cell, a skin cell, etc.
During G1, the DNA of the cell is in the nucleus in the shape of a double helix. Next, synthesis or S phase. In this phase, all the cell does is duplicate its DNA. The final stage of interphase is G2. The cell finishes growing and does its normal job. The cell also makes two centrosomes to be used later.
There are 5 stages in mitosis. First, prophase. In prophase, the cell moves the centrosomes away from each other. The nucleus starts to disintegrate as well. Second, prometaphase. During prometaphase, the centrosomes with the spindle fibers are at the opposite sides of the cell. The nucleus and the nuclear membrane have almost disappeared at this point.
Third, the cell goes through metaphase. This is where the chromosomes lie up at the center of the cell and the spindle fibers attach to the centromeres of the chromosomes. Fourth, anaphase. This is when chromosomes split and have the spindle fibers attached to the centromeres. Next, telophase. In telophase, the cell starts breaking up into two nuclear membranes, starting to form around the separated chromatids. In cytokinesis, the cell actually breaks off into two cells, creating identical offspring to each other in the parent cell. These cells are 2N and have chromosomes that were produced by asexual reproduction.
There's meiosis 1 and meiosis 2. Meiosis produces 4 new cells from the single parent. During meiosis 1, crossing over happens. This is where parts of the chromosome switch places to create genetically different cells. The switch is random. In meiosis 1, there are 5 stages. First, prophase 1. During this phase, the chromosomes attach to each other and the nucleus starts to disintegrate. In prometaphase 1, the chromatids that are attached to each other twist. The centrosomes are also on opposite sides of the cell. During metaphase 1, the spindle fibers attach to the centromeres of the chromosomes that are lined up in the middle of the cell. In anaphase 1, the chromosomes are being pulled apart by the spindle fibers. This plus the twisted chromatids causes the DNA to switch a part of the DNA.
In telophase 1, the cells start breaking apart. The cells at this point are 2N. Next comes meiosis 2. Prophase 2 happens, where new centrosomes and spindle fibers form. Then, prometaphase 2 happens, causing the spindle fibers to attach to the centromeres of the chromosomes. After that, the chromosomes line up along the middle of the cells. Anaphase 2 then takes place, where half of the chromatids move to each side of the cell. Then telophase 2 happens, where each cell breaks up into 2, making 4 gametes or 1N cells. 2 gametes can come together to form a 2N cell called a zygote, which is the basis for sexual reproduction. This is why meiosis is important.
There are differences and similarities between mitosis and meiosis. A cell in mitosis creates 2 cells, where a cell in meiosis creates 4 cells. Meiosis is sexual even though there is one parent cell.
Mitosis is asexual. Meiosis creates gametes, which are 1N, although mitosis creates normal 2N cells.
Some similarities are that they both have their steps starting with the same 5 words. Both mitosis and meiosis have one parent cell and both happen in cells and create more cells.